Well, well, Mr. Anderson.
Mr. Smith, good to see you again.
You have run away for quite some time now.
Yeah, and I've escaped too. Just give up, Mr. Smith.
No, not this time. I've got you now, Mr. Anderson. PMAT! Pro! Meta! Anna! Tele! Phase! Mr. Anderson, you can never hide from me. You can never hide or run away from me, Mr. Anderson.
Smith, I don't understand. I haven't gotten caught by you ever. How did you catch me this time?
Well, what I did was similar to a lesson I learned in biology class.
What did you learn that helped you finally catch me?
The process of mitosis.
How does that work?
Well, I learned that mitosis is the process of dividing the nucleus into two parts for creating offspring cells consisting of 4 stages: prophase, metaphase, anaphase, and telophase.
Oh, I think I've heard of this in my bio class. Anyway, go on.
Well, prophase is the stage in which centrosomes containing centrioles move in opposite directions inside the cell. At the same time, the nuclear membrane disappears and the copies of the chromosomes, the chromatids, are connected by centromeres.
In metaphase, the chromosomes intersect and string tight.
Indeed, the chromosomes attach to kinetochore fibers which were made in prophase and extend from one of the centrosomes to the other.
Wait. Do you have pictures? I mean, it's kind of hard to see.
Well, I always carry my pictures around.
Can I see? What have we found so far?
Well, here we already have prophase, and now we're up to metaphase.
Okay, so prophase. So we have centrosome, nuclear membrane. Oh, okay, and the chromatids, they've duplicated, right?
Yeah.
Yes. Okay, I understand that. That's good.
Now we have metaphase, which I explained. The chromosomes attached to the kinetochore fibers, as shown in this picture.
Okay, so right here, basically, they're all lined up in the middle, right? And these things, these like lines, they're getting ready to split them apart.
Yeah.
Okay.
Exactly. There are also polar fibers which extend across the cell too, like the kinetochore fibers, but do not attach to the chromosomes.
Oh, wait, I remember. Now, the stage anaphase is when the chromosomes are split and pulled apart by the what's the word?
Kinetochore fibers.
Kinetochore fibers, and pulled into each centrosome. This way, each copy of the cell's DNA is identically and evenly distributed to both sides, right? Is that
Absolutely right, Mr. Anderson.
do you have a
It's right here.
awesome. Okay, so we're seeing right here like they're this is anaphase where they're going like splitting onto separate sides like that. Oh my God, this is so great. Oh, that's how. Okay, so is there anything else or?
Well, there is the last stage of mitosis, telophase. In telophase, the spindle fibers disassemble and the chromosomes on each side of the dividing cell are then enveloped by their own nuclear membranes.
Wait, how does a cell split into 2 new cells?
Well, that's another process called cytokinesis.
Cytokinesis?
Yes,
Cytokinesis.
After the DNA has successfully divided, a cleavage furrow begins to form in the middle of the cell, in which microfilaments are pinching inward in the cell membrane, and eventually the cell splits into 2.
Do you have anything I can see? I need a better.
No,
Awesome. These are very good last words. Alright, so what I'm seeing here, the cleavage furrow right here, I see this. They're splitting into 2. Nuclear membranes are reforming.
Exactly.
They're going to even side. I understand this Thank you, Mr. Smith, for giving me this lesson before you annihilate me here. Let me just I'll keep that for yeah.
Well, that's the end of the lesson. Now, it has brought you to your doom.
Haha, Mr. Smith. You didn't think us out. You just taught me how to reproduce, how to double, PMAT, prophase, metaphase, anaphase, telophase.